Thank you for coming today. I am Kirsten Lyke, and I am thrilled to present an introduction of David Rach and his thesis defense today. And Cristiana and I will be toggling back and forth.
Thank you, everyone. So I'll start introducing David. Child of the board, he was raised in Mexico, in Chiapas, nearby Guatemala. So his early years were very colorful. His first language is actually Spanish, or at least for a period of his life, his first language was Spanish. He enjoyed that period of his life. But at some point, since he's a very curious person and he likes the snow, he decided that he wanted to study in Wyoming. And so he attended college at Northwest, and then he obtained his Bachelor in Science at the University of Wyoming. And then since he's kind of restless sometimes, he decided to go serve in the Peace Corps, and he's always been interested in global health. So he decided to serve in the Peace Corps for 2 years in Ghana. And another one of his passions is teaching. So in those 2 years in Ghana, he was teaching at Clicker Secondary Technical School, biology and chemistry. And you see the blackboard there. That blackboard is pretty similar to the whiteboards that get filled up with scribbles and figures when they discuss spectral flow cytometry, and in particular, autofluorescence. You will hear quite a bit about that later. So after his 2 years in the Peace Corps, he decided to join the University of Maryland. He joined the University of Maryland MMI program in 2018. Then in 2019, because of his interest in global health again, he joined Kirsten and I. We had a project in need of a student, and he was curious about global health. And the project was meant to investigate the impact of HIV infection during pregnancy on the development of the fetal immune system. So he joined us in late 2019. He passed his qualifying exams in February 2020. And then we all know what happened. COVID happened. And Kirsten will continue.
So COVID, as it did for all of us, derailed the plans. David, making lemonade out of lemons, joined the Pfizer-BioNTech study. If you look up the first person to have received the Pfizer-BioNTech vaccine, they'll talk about a nurse who received the first licensed. But David was the first person in the world to receive the Pfizer-BioNTech vaccine. So we were derailed in terms of the clinical study. It took a little bit longer to get the cord blood and PBMCs to our center. And then we made a switch from mass cytometry to spectral flow cytometry, which was really in its infancy when we began, unbeknownst to us. So what began as an immunologic study looking at maternal fetal pairs with HIV became a deep dive into spectral flow data analysis. And I think we've identified David's true passion through this. And as you know, he's really taken this on. There were a lot of holes in how to analyze the data. And he, rather than just giving up or using an app that was perhaps not fully flushed out, he made his own apps and now has 3 R packages on GitHub. And then if that was not enough, he has this past year organized cytometry in R, which has hundreds of people online and an in-person study for a 30-week course. We're in week 11, I believe. And outside of work, David has a passion for birding. You might recognize some of the titles of his apps. For outdoors, for kayaking, for ultramarathons, all fueled by coffee, copious quantities of coffee, and supported by the people and animals he loves. So with that, David, at long last, in many ways, welcome.
All right. Thank you, Chris. Thank you, Chris, and thank you, Kirsten, for that introduction. So hello, everyone. For those of you who don't know me, my name is David Rach. I'm a PhD student in molecular microbiology and immunology here at the University of Maryland in Baltimore. And welcome finally to my thesis dissertation, where I'm talking about characterization of cord blood innate-like T cells in neonates born to healthy women and women living with HIV. So if we step back and think about what the immune system is actually boils down to, an immune system recognizes self from non-self. So if you think in context of like microbiota in either our gut or our skin, they stay relatively separated from self. So there's not actually much of an immune response that's happening on a day-to-day basis. However, if you have a breach in the barrier and all of a sudden you have the self and non-self mixing together, the combination of both danger and damage signals in the presence of non-self means you initiate an immune activation. And then after the situation has been resolved, the immune system, the cells that have responded to the threat, end up a few of them persisting, and these become memory cells that are then able to more rapidly respond to threats in the future. So if we think about this paradigm, but then we apply it to fetal maternal immunology, specifically in the context of pregnancy, the maternal immune system, how does it view the fetus? So it still sees the fetus as 50% mom, so that is being recognized as self, but 50% of it's being recognized as non-self originating from the dad. So the question is, does the maternal immune system respond to it? Well, that depends. Is there interactions going on between self and non-self? And is there any danger or damage signals being produced that may lead to an interaction? So if we think about it in context of neonatal immunology, the fetal-maternal separation is mostly in place through the placental barrier. There's still a few cells that get through from mom to fetus and vice versa, but for the most part, cells remain on their sides of the respective part of the interface. At the same time, nutrients can go in, waste can go out, as well as maternal antibodies that end up being transferred before birth. This is very important during the early life period because as the baby is born, they start entering this brand new world of microbial exposures, and they need to decide how exactly do we react to things? Are we reacting to everything as a threat? Are we reacting to everything as a non-threat? This balance happens during the first 6 months of life, and the maternal antibodies that are transferred both by the placenta and through breast milk slowly wane. And then as the infant immune system slowly stands up and responds in kind as far as what we would expect for an adult immune system. So for those of you who are not immunologists, if we step back and think during this early life period, most of the immune cells that they would need at that point already are present. They're just not yet exposed to pathogen. They're not yet differentiated in terms of their antigens. So the very simple flyby view of immunology, we have innate-like cells, things like dendritic cells, monocytes, granulocytes, and K cells that respond rapidly when they sense a threat. And they're recognizing as far as what's being seen. Vice versa, there's more the adaptive immune system, things like B cells and T cells, which have a slower response time, but they're antigen specific, meaning they can target specifically proteins or other biological molecules present on those individual cells. And for our lab, one thing that's of particular interest to us is innate-like T cells. These include natural killer T cells, so things that would be staining for with Vα24, J-alpha-18 on the antibody side, or if we're using tetramers, CD1D loaded with something like PBS57, which is a lipid. Vice versa, we also have mucosal-associated invariant cells, things that would be staining for Vα7.2 and also be CD161 high. Or if we're using our tetramer method of characterizing them, they're MR1 tetramer loaded with APRU56, which is one of those vitamin A metabolites. And then we also have the Bγ9 Vδ2 T cells, some of the gamma deltas in general, but the ones that are more innate-like in nature. And we're generally just staining those in context of our panel with the Vδ2 antibody. So innate-like T cells are important during this early life period because they bridge both the innate and the adaptive immune system. They refuse to be boxed into the little categories we have developed as far as the innate and the adaptive. They're able to activate both in response to their antigens, but also in response to cytokines from the general signal, much more similar to what those innate immune cells would be doing. And they're particularly important in early life because they are already been shown by other papers to already be producing cytokines at the time of birth. And they are thought to be a bridge as far as like during that early life period when the adaptive immune system is not yet ready to go to be able to stand in and carry out the role of protection in case of an infection. Which brings us back to the paradigm of what's happening during pregnancy. So if you have an infection during pregnancy, we had left off with the question of, does the maternal immune system respond to the fetus? And we had left it at that point of, is there an interaction or is there damage? So obviously, if the mom has an infection, she's going to respond to that infection. It's not just going to be left with free rein to cause damage across the board. So at that point, the maternal immune system is having calls to arms. There's inflammation, there's activating cytokines roaming around the system. There's T cells and B cells and all the different innate cells searching for their targets to be able to eliminate them. Now, all the activation at arms from the maternal side end up impacting the developing fetal immune system on the other side of the placental barrier? So we're not talking a congenital infection where the babies themselves are infected. We're just talking something on the maternal side. Obviously, at this point, it depends on location, scale, and timing. So if the mom has an upper nasal infection, that's just like a small sniffle, the odds are less likely that that's going to impact the fetus on the other side of the placental barrier. However, if the mom has something like influenza and systemic and its entire body reacting to it, it's much likelier that those activated cytokines that are running around the bloodstream, because it's being shared with fetal circulation, will cross it and will have the potential of impacting the infant immune system. So what effect does this early exposure have on fetal immune systems? Well, there can be multiple options, and this is where I'm going to switch to my pointer if I can. Maybe not. I will highlight with the hand. So there's a situation where that maybe there's no activation of any form happening on that side of the placental barrier. Maybe the cells just do not know there's a war going on in the maternal side, and they just remain perfectly normal in terms of their trajectories of differentiation as they get ready for time of birth. On the flip side, maybe the infant immune system, the fetal immune system, reacts exactly like the mom's immune system to these threats and starts arming up and starts differentiating out. And then it becomes a different, more advanced phase than what we would have expected at that time in the pregnancy. Vice versa, there's a lot of immunology that's trying to tamper down on inflammation just to make sure the pregnancy is successful. So maybe as the fetal immune cells start getting activated, they end up getting repressed to be able to prevent that from happening. Or maybe they activate too much and they are targeted for deletion and they're just gone entirely at the time of birth. So if we're thinking about after the pregnancy, during that early life period, as the infants are exposed to the first set of both commensal as well as their first infections, how does these potential exposures early in life set them up for success or set them up at increased risk of being seen at the clinics? So we can hypothesize like all these different scenarios, like depending on what we have, how this might turn into a certain outcome. And this is not just a theoretical exercise, so it's actually of importance when it comes to HIV-exposed uninfected infants. So HEU infants are born to women who are living with HIV. They have been exposed to maternal viral load and proteins during the context of the pregnancy because they are seroreactive to those proteins, but they are not infected by the virus. So this is good because we don't want the babies to have HIV infection. But interestingly enough, during their first 6 years of life, they are seen at much higher rates in the clinics for lower respiratory tract infections as well as diarrheal diseases compared to their unexposed counterparts who live in those exact same communities. Now, as immunologists, we always want to find an immunological cause. So we see a pattern in the data. We try to figure out how exactly it works to be able to better explain what we're seeing. So as an immunologist, our first instinct is like, well, we know there's potential for impact during fetal exposure. Maybe the cells are too active and they're just going overboard when they see their first commensal bacteria, which is why they're ending up in the hospital. Maybe the cells are inactive and then the bacteria is just causing more rampant damage. Or maybe the cells that are needed to target the certain situation are just missing entirely. Now, there has been other immunological studies looking at these HIV-exposed uninfected infants because we've been seeing the same kind of clinical pattern dating back to the early 2000s. Some of the studies have shown that the T cells have increased frequencies of Th1-like cytokines being produced ex vivo. Some of them showed decreased proliferation in response to culture right after birth. And then others have shown that there's been a lowered amount of maternal antibodies being transferred. But at the same time, while 10 of the studies show these kind of patterns, another 15 show there's no difference, or it's actually going up as far as maternally transferred antibodies. So as far as trying to figure out what is going on with these infants, attributing a cause, it's much harder to parcel out when you're weighing the evidence as far as these studies. As much as we may want everything to be an immunological explanation, there could also be socioeconomic factors. So if you think about it in context of the infants, maybe this individual studies differed as far as like, were the moms breastfeeding or were they formula feeding, which at one point was a policy recommendation. Or maybe there's a difference when in terms of the maternal viral or maternal antiretroviral exposure. So if you think about this in the context of a policy timeline, right at the early part of the HIV pandemic, antiretrovirals were really hard to come by in lower middle income countries. So if you got them at all during pregnancy, the mom was given the antiretroviral right before birth and then immediately taken off the antiretroviral after the baby was born. So mom still has HIV, she's still viremic during the early life of the infant. So that's a potential cause of exposure. Vice versa, as things have improved in terms of the antiretroviral access in lower middle income countries, mom, as soon as diagnosis, was put on either individual antiretrovirals or now in the present day, combination ART for the duration of life. So if we look at the conflicts between the previous studies in terms of is there differences being seen or are they not? It could be they're still treating HIV-exposed uninfected infants as a broad category, but within that broader umbrella, they have infants whose moms knew their status, were on ART, had undetectable viral load, and therefore very little viral exposure. Or they also had moms who had only found out shortly before pregnancy and were still viremic, in which case there was a lot of exposure going on at that point in time. So the University of Maryland, especially the Center for Vaccine Development, has been working closely with partners in Malawi for over 2 decades now, originally with the Blantyre Malaria Project, more recently with the CUHES, as far as the affiliation changed. Ultimately, what we've been able to do, because Malawi has such a high HIV burden, is a lot of the people who are coming into those clinics are women who are living with HIV. So as part of the broader people that we serve, the study was instituted as far as trying to puzzle out what effect does this viral exposure have versus what effect could be attributed to antiretrovirals, which are known to interact with mitochondrial pathways, which can similarly cause low-level inflammation. So what we ended up doing for our cohort stratification is we enrolled moms who did not have HIV, at which point their infants would have had no viral and no ART exposure. We also enrolled moms who knew their status well before conception, had undetectable viral load throughout the pregnancy, but would have had continuous ART exposure throughout, as well as moms who only found out their HIV status during the first antenatal care visit, which typically is around 4 months into the pregnancy, at which point half the pregnancy has basically gone by with high viral exposure, but only then antiretrovirals start up for the second half. And then as far as the study timeline, the main focus of what I'm showing today is focusing in on the cord blood as far as the specimen goes. But the study broader goals also took 4 and 9-month peripheral blood draws so that we could monitor how the vaccine responses from these infants change over time in response to these different exposures. So even in the most carefully controlled, groundbreaking study at the time are defined by the technology and the resources that those studies had available. So if we think back to those early 2000 studies of HIV-exposed uninfected infants, many of those were done on like a fax caliber that could only look at 4 fluorophores in terms of flow cytometry markers. More recently, a lot of the studies that were conducted in the middle 2000s were done with the LSR2, which could handle up to 14 if you had it fully spec'd out more on the 18 side as far as total markers that can be profiled at one point in time. In context, if you think nowadays with spectral flow cytometry, it's much easier to look at 30, 40, or as we saw recently at the CITO conference, up to 60 fluorophores in a well-designed panel. So you're able to profile a whole lot more markers than what we would have been able to do at the time. That's not a discredit to those studies. The studies were still groundbreaking, but we are narrowed down on our resolution as far as what we can do to puzzle apart this knot based on what's already there. And this is part of the reason why there's very little that's known about cord blood ILTs. So there's been a couple papers about gamma deltas. There's one or two papers in general for MAITs and NKTs. But the reason for this is this cord blood ILT subsets are kind of hard to piece out with the technology that we had at the time. So because they are unique, they require additional lineage markers like Vδ2, Vα 24, Jα 18, etc. Which made it not particularly feasible to include within a conventional flow cytometry panel as a side project. It would have to be like the main focus of your panel to be able to actually get any characterization of them. At the same time, cord blood ILTs are relatively rare. They're less than 1% of the total T cell population in cord blood. So even though mass cytometry has been around for much longer and is able to profile extensively the breadth of markers, it would have required significant amount of runtime, especially on those early mass cytometers, to acquire the 3 million events to be able to see the just 100 cells that are actually present. So in the context of this population, it's one of these things where it wasn't quite grasped by the technology at the time. Which is where we come to the changes in flow cytometry. So regardless if we're talking the older conventional flow cytometry or the newer spectral flow cytometry, they are primarily focused on characterizing two things about your cells of interest. They want to know on a cell what fluorophores are present or not present, and then in what amount are the fluorophores that are present there at. So conventional flow cytometry operated under a one fluorophore, one detector paradigm. Basically, any of the signal that was coming in on this peak detector was attributed to belong to Ph.D or to APC. And anything that was also spilling over that's present lower down was just mathematically subtracted out as far as the contribution. So that way we could say if we see signal on the Ph.D detector, it must be coming from whatever antibodies bound to Ph.D. This meant that if there was fluorophores that are similar but have extensive amount of overlap, they cannot go into the same panel, which means the overall conventional flow cytometry panels are much smaller. Spectral flow cytometry comes at this problem from a completely different approach. It's a very similar technology. It has a lot of the moving parts, but it's its own beast when it comes to interpreting out what is there and what amount. So the way this basically works is those unmixing controls provide reference signatures that define what an APC and what a Ph.D looks like. In turn, taking from the full stain sample, it does a unmixing, which is kind of like a deconvolution of the individual components that must be there and in what amount using linear algebra. The end result of this technology is that you can combine fluorophores that are extremely overlapped with each other and still be able to adequately resolve out the difference in the end. And both conventional and spectral have rapid acquisition speed, so acquiring 3 million cells in a matter of a couple minutes is feasible. So as we entered the bright new world after 2020, and we ended up switching to spectral flow cytometry, our initial response was trying to treat spectral flow cytometry in the exact same way we had treated conventional, where we acquire the data, we try to compensate it in FlowJo until it looks just about right, and we call it a day. But it was being very stubborn and was not cooperating with what we were trying to shoehorn it into. And the reason is there's several pink elephants in the room with us as we speak for spectral flow cytometry that we were just not aware of, or we didn't appreciate the value of quite to the extent that we needed at that point in time. So one of these is that the quality of the unmixed full stain data that you get out that you actually want for your experiments is inextricably linked to the quality of the unmixing controls and the autofluorescence stand-in that you are providing to the system. And I'll talk about that in a little bit. But then also, if you think about spectral flow cytometry, it's its own unique beast. And this ability to not only acquire a lot of markers, but a lot of cells in a relatively short amount of time means the data sets that we're acquiring are substantially larger than what was expected for conventional flow and mass cytometry panels back in the time. So the algorithms that were supposed to handle the data were just not up to par and would crash out due to lack of RAM. So I've mentioned spectral signature a couple times. Here's a couple examples. So, for example, P-Vial 770 is on a yellow-green laser where you see the max emission. You can see it has a couple peaks versus its tandem counterpart, APC-Vial 770. You see there's just more of a single peak that's happening over in the red. Now, even to this day, I don't remember all the fluorophore signatures. There's over 1,000 fluorophores. It's just not possible. My brain's too focused on other things. But in terms of explaining how this unmixing process works behind the scenes, I find using boxes is a better analogy. So let's think if we took that signature for APC vial and broke it down and made it just a unique kind of fingerprint that your instrument's recognizing and doing all the hard math behind the scenes for. So APC vial, let's say the computer can recognize it as two pink blocks stacked together is equal to an APC vial fluorophore. And then vice versa for your P vial, it's two green blocks stacked together is equal to the P vial one. So what's actually happening is rather than trying to compensate out spillover, it's unmixing on a spectral flow cytometer works by looking at individual cells, seeing what the totality of the signal is, and then trying to like deconvolute and figure out what actual contributions must be there. So in the case of like APC vial, we can see there's two pinks. There's at least 4 pinks in this total full stain cell. So there must be about 2 of those present on the surface of that cell. Vice versa, the little blue fluorophore has 3 blocks of blue. We don't have any blue blocks on the surface of that full stain, so we're able to infer that it's not present on that individual cell. And fortunately for us, we don't have to repeat this manually for all however many markers are present in our panel. The linear algebra behind it is pretty solid as far as the approximations go. So this is how we are able to basically infer what is present on the surface of the cell and in relatively what amounts. So what happens though when those unmixing controls, which we are relying on to give us accurate measurements of the underlying data, end up being corrupted or not actually up to par to where they're supposed to be? So this can happen for multiple reasons. Fluorophores are biological molecules. They can get damaged by reaction to oxygen species, which changes their signature. Also, we have autofluorescence in our system, but as anybody who's had the misfortune of working with myeloid cells will attest, they can have a lot of different autofluorescences for a lot of different molecules. So maybe it's not just one in your system, maybe you have 5. So at that point, the assumptions as far as the math go are off because you're not actually accurately determining things. So for our little toy example, let's say this little brown fluorophore ends up having a little bit of a mistake, ends up having a little bit of a green signal incorporated into its reference signature. At that point, you can see downstream at the time of unmixing, the amount of brown is actually being uncalculated, lower. But then what we had estimated to be present for green has been completely obliterated just because its contribution has been commandeered for something else. And this is what causes the unmixing errors that we encounter with spectral flow cytometry. These are distinct as far as like forming blebs, having a risk appearing back to the negative appearance. And these are particularly problematic, especially in the realm of unsupervised analysis, because the unsupervised analysis algorithms are predicated on having a reliable start and end when it comes to the MFI measurements that are being seen. So not only is this making it harder for you when you're manually gating on a supervised kind of workflow, it's basically taking a lot of the work that you could have done through use of an algorithm off the table from the get-go just due to data quality. And more concerning than anything else is, if you're looking at this, sometimes it's not quite as obvious. And then that question comes in, is the population that you're seeing actually biological and actually of interest, or is it just a technical artifact that's being manifested because of improper unmixing controls? So why does this matter? So while spectral flow cytometry is great, it also gives you the power to shoot yourself in the foot, which is what we saw for one of the papers I presented in Journal Club several years ago, where they ended up using the same viability dye as their main marker of interest in the same panel because they had slightly different names, at which point I can't actually know what that paper actually is publishing, whether it's their main cell population of interest that they're making all the claims for, or if it's just they're looking at the most necrotic cells in their system. But overall, this is just more emblematic of oftentimes we try to treat something new as, oh, it must be a continuation of the old, let's just optimize it a little more, when in reality, we should probably start and try to understand the fundamentals of what's going on before diving headfirst into a empty pool. So as in always, everything with science and everything with algorithms, if you put garbage in, you're going to get garbage out. And more concerning on my end, and this is my little soapbox of the day, you're adding to the reproducibility crisis within science. And at the same time, you're not only wasting your time, your grad students' times, your postdocs' times, your PI's times, you're also losing all that effort and all the funds that went into all those failed experiments that you would then need to repeat. So, if anybody has ever met me, they know I really do like puzzles. I've loved puzzles since I was like 2. There's something about like putting together the small pieces, building to a bigger picture that's just satisfying about puzzles, which is why our family loves them. So at that point in time, I had things about this mixing process that just were not clear for me. The papers that actually would have explained it were several years still in the making. So at that point in time, my main concern was how do I go about trying to figure out how the system actually works? And how do I try to figure out a better way to deal with these unmixing issues that impact our data quality without spending an inordinate amount of time compensating everything back and forth for 6 hours on a per sample basis? So ultimately, at the central core of this problem is reference signatures from your mixing controls are critical to our final quality of our data. And despite this critical importance, there were no tools available to screen signatures before the fact to see what impact they would have on a mixing. This was a common community recommendation to the vendors. The vendors just didn't get around to implementing it. As a result, the community as a whole would spend hours trying to figure it out on their own using less precise methods. But at that point in time, thinking about the things that can affect the fluorophore, they're going to be changing the signature. And whenever there's a change happening in the background, there's always some trace that's left over. So when we've been characterizing signatures, we most of the time are looking at an entire population, taking the average, and then using that to generate the signature. But at one point in early 2020, late 2022, I had a little moment of insight. And I was like, what if instead of trying to look at the average of this population for the signature, we tried to quantify it at the individual cell level? And this was still not fully fleshed out, but it ended up sparking a actual move that if you had asked me at the beginning of my PhD, I would have told you were crazy. And this is basically starting my journey into learning R programming. I was not anything close to computational at any point before I showed up here, and I was not anything close to computational for my first 3 years of my PhD. However, when it comes to trying to figure out and making sure I get the best quality data, especially given this unique cohort, I chased this rabbit down the rabbit hole and ended up trying to figure out how to build my own tools to be able to get at the situation. And a lot of this work dated back to that early phase from 2022 to early 2024 when the actual Luciana package got created and was put available on GitHub. And basically the way my Luciana package works is it figures out what the individual signature is for these individual cells at that level, and then just ends up grouping cells that have similar peaks of similar heights together, and then taking these groups, and because I have figured out how to do the FCS internals, generate out a fully compatible FCS file that I can use in SpectraFlow for unmixing after the fact. It has also had a lot of benefits when it comes to automated methods of screening our mixing controls, where I don't have to think about it. I hit run on the pipeline, and it's a 15-minute process to figure out what could have gone wrong for a given experiment. So here's a couple of my favorite examples. APC Fire A10 can degrade back down into the APC fluorophore, in which case the recommendation is we need to get rid of that vial of antibody and buy a new one. We also see something similar that happens more on an experiment basis, where whatever you did to your cells during the processing generated reactive oxygen species that also damaged the tandem fluorophore in that individual tube, in which case the recommendation is replace the tube. And then we found things that are a little bit harder to diagnose as far as like residual autofluorescence contamination to the single color controls, which was something the community was not quite as aware of before this, as well as a way to isolate out autofluorescences in a relatively easy manner that we can then use to test for unmixing. So overall, this was a dramatic improvement to our data quality, where the point where it was actually feasible to even consider going the unsupervised analysis route, because at that point, the algorithms would perform similar to what they would have done for conventional, but also what they would have done for mass cytometry, which doesn't have the autofluorescence, which doesn't have as much of the spread. However, we still have one pink elephant left in the room, which I'm just going to touch on briefly for now. Namely, we have a lot more cells than what those previous algorithms were able to handle. So how do we go about trying to analyze it all, especially in the context of not just looking at my tiny population of cord blood ILTs, but trying to figure out what's happening in the rest of the sample? So if we step back and think about this, if for every marker in your panel, everything divides into either a positive or a negative for a given marker, CD3 positive, CD3 negative, and you repeat the splitting process for every single marker in your panel, and then at the very end, just group things together that share the exact same expression of every single marker. This is the exponential climb as far as potential groupings that are present in a given panel. So for a 9-color conventional flow cytometry panel, you may have only had 512 of these groups at most, doing 2 raised to the number of markers in your panel. A little bit there, but your grad student could definitely have gotten that done over a weekend, so it's much more achievable and much able to do. However, if you're thinking of our 40-color panel, there's over 1 trillion of these potential groupings. And if you're thinking of the more recent 60-color panels that were announced at CITO last week, that's over 1 quintillion potential groupings of cells in the high-dimensional space. Now, fortunately, biology is not math, thank goodness for that. But at the same time, there's still a lot more than what a supervised manual analysis would actually be able to implement for the analysis. So if you're thinking unsupervised algorithms, most of the community is currently using things like dimensionality visualization, dimensionality reduction, things like UMAP, TISNE, FATE, PACMAP, as well as different clustering algorithms like FlowSum, PhenoGraph, XShift, etc. So these are trying to make similar kind of splits in the biology based on different algorithmic metrics, generally based on MFI. And they sometimes work, they sometimes don't. And it's often a point of discussion within our community. And the reason for this is at the far ends, we all agree what makes up a cell, but then when you start doing the nitty gritty on a basis of an individual marker, it will vary a bit. So the community as a whole agrees if you're running a clustering algorithm and all your cells end up in their own individual clusters with just each other, at that point, that's clearly not right. On the far end of the spectrum, when all the cells end up in a single solid cluster, we also know that is not correct. But all the arguments about which algorithm is better than the other is just basically where in this continuum in the middle are they actually causing splits? Are they grouping cells that are different from each other together into a single cluster? Are they splitting what's normally a single cell population into multiple cell populations based off of just a little bit difference of MFI? This is where the field is as far as trying to figure out what is the best implementation to deal with this avalanche of data that we have heading towards us. So I'm putting a pin as far as what I ended up doing for my approach to this, but I'll circle back and propose not necessarily a compromise, but something that seems to work well, at least in the context of what we were doing. So at that point in time, having fixed our unmixing issues, our previous largest panel that we had for the Cairo lab at that time was an 11th floor on the LSR. So we still needed to optimize up to what eventually became our 29-color spectral flow cytometry panel. And this included all the lineage markers we would have needed for the ILTs. It included phenotypic markers for both T cells and NK cells so that we could snoop on what they were doing in addition to what we were looking at our ILTs. We included a couple cytokines, TNF-α and interferon gamma. I tried a CD107A that was so-so for this first panel iteration, but then also because I was interested in MAITs and NKTs and had been reading literature about difference between antibody staining versus tetramer staining, I included a way of, in the same panel, switching out the antibody for the tetramer to see if I saw any differences. At that point, it was fairly typical for a flow cytometry processing of the data. I would thaw the cryopreserved specimens, I would count them, I would aliquot them. I did PMA ionomycin staining for 6 hours just to activate them broadly across the board, at which point it entered the flow cytometry staining, which basically consisted of a surface stain, the tetramer stain, an intracellular stain. All in all, this was an 18-hour day for each single experiment. So by the end, I was tired and I just wanted to go home. So I ended up acquiring the data the following day as far as the actual acquisition of the cells, at which point in time, the Cytec 5 laser Aurora, thanks to Neville, was already upstairs. So I was able to use it with much better quality data than if we had been using the 4 laser. And then at that point, we had really good data and resolution on our populations of interest, as well as additional ones that might be of interest for general public. So in terms of the actual specimens I ended up processing, the good news is if you think back when I mentioned most of those HIV-exposed uninfected high infants, their moms found out their status halfway through the pregnancy. By the time they actually gave birth, 80% of these moms had already cleared the viral load from their bloodstream and were undetectable at that point in time. So the ART seems to have kicked in and done its job for the vast majority of those mothers. And then at the same time, we know that sex-specific differences and gestational age can impact what we see for the immune cell populations. So we tried to make sure that for the specimens we were acquiring on our spectral flow cytometry panel, that they were balanced in terms of across these different exposure groups. And as we suspected, ILTs are indeed rare in cord blood mononuclear cells. So Vδ2s were the more abundant of the cord blood ILT subsets, but there were still only about 1% of total T cells within a cord blood specimen. MAITs and NKTs were much rarer, but we still saw them in every single specimen across the board. Interestingly, MAITs, as far as when we're staining with antibody, had a greater percentage as compared to when we were doing MAITs by tetramer staining, which I always found interesting, especially because we didn't see the same pattern for NKTs. When we were looking across the exposure groups, we found that when you combine the HU low and the HU high, we see a lower amount of MAITs and NKTs as far as the frequency goes in these HIV-exposed uninfected infants. However, we only saw that in one condition of the different ways we tested. So we saw it in the antibody condition for the MAITs, but not in the equivalent tetra condition. And we saw it in the NKTs, but not in the antibody condition for NKTs. So it's there, it's present, but how strong of a conclusion we have from this one is something I'm still mulling. Additionally, we also are characterizing cord blood ILTs in ways that were not possible previously. So we wanted to get a good baseline of a larger number of markers, just because this is data that's good to have for the community that we didn't have previously. So memory differentiation was one of those things I first tackled. So all the different ILT subsets are different in terms of their memory markers. So NKTs are the more differentiated of the bunch at birth. MAITs are mostly naive at this point in time, and the Vδ2s are somewhere in the middle. As much as we looked, there was no obvious difference in memory differentiation in terms of the HIV-exposed and infected infants. So of those 4 different potential scenarios we had back in the graphical abstract, we can cancel that one off. We don't see a difference on the memory differentiation front. Additionally, we were curious about the cytokine production. So when we checked like the PMA inomycin activated samples, we saw that in general, the cord blood ILTs are making cytokines similar to the frequency of what we would see for like the NK cells. So those more innate-like cells, which makes sense considering the adaptive-like T cells are not yet up and running. They're not being exposed to antigen. They're not as differentiated as we would expect. So, for example, the Vδ2s and NKTs made a lot of TNF-α, as did the MAITs. Interfering gamma was primarily a Vδ2, as far as like about 50% of the Vδ2s had interfering gamma after stimulation, which is still lower than what we saw for the NKs, which were around 75%. And then vice versa, those that are polyfunctional for both TNF-α and interfering gamma, it was the Vδ2 and the NKTs that had about 25% of their cells falling within that cytokine production category. And what I found absolutely fascinating, given that MAITs by tetramer staining were a lower frequency than what we saw for MAITs by antibody staining, and based on previous papers published by the Swarbrick group, I saw there was no statistically significant difference in terms of the marker expression for any of the markers in our panel for either MAITs when stained with antibody or with tetramer, or for NKTs when they were stained with antibody or with tetramer. So as far as what the difference of our study is versus the other study, I don't actually have their data to be able to check, but my suspicion is we were probably gating in a different way from what they were. So maybe they were including additional cell subsets that our lineage definition of MAITs of HMR1 positive, CD161 high, maybe was being a little bit more restrictive as far as what we're seeing, and we're just isolating out the pool that would have been tetramer specific to begin with. And at that point, once again, we're trying to do is a general atlas of what we end up seeing. So we're taking advantage of what we have. I just wanted to characterize broadly for the 3 cord blood ILT subsets. So Immunologists will go have a field day with all the different markers. We have 29 different markers. I'm not going to go through all of them. I'm just going to show you the highlights as far as like the individual subsets. So what struck me about the mates was primarily mates are all CD26, CCR6, CD161. Most of the cells are having expression of those markers. And as we already mentioned, most of them are naive. And one thing to note for CD62 ligand, it's not really following what we see for the CD4, CD8s, at least in the context of cord blood ILTs. Vice versa, when we activate our mates in the cord blood, we end up seeing that CD69 pops right up as far as the activation marker. So our PMA ionomycin is working as we were intending. But then it seems to be mostly TNF-alpha that's getting produced by the mates at this point in time. As we look at NKTs at baseline, you can see the pattern has changed. One thing I am definitely adamant about is in our data, looking at all our NKTs, we do not see CD56 and we do not see NKG2D. So all the global health papers that have come before us claiming their CD3 positive, CD56 positive population is NKTs. Just like others in the community have pointed out, this is not actually the case. These are different cells, probably more on a TNK-like kind of population, not the actual type 2 NKTs in this case. And then NKTs, as we already mentioned, are more memory differentiated, and they do have a good amount of expression of both CCR4 and CD25 across the board. And then when you activate them, just like everything else, they get a lot of CD69 activation. There's a lot of TNF-alpha, and there's a good amount of interfering gamma on the lower end, although not quite as much as what other studies have reported, which could be they're not getting the right kind of cytokine help signal that the other studies had in culture settings. And then for cord Vδ 2s, it was kind of a blurry as far as the initial results looking at the global population. So everything seemed to have CD27, but if you look at this particular marker expression plots, they're all kind of like averaged out somewhere between like 30 and 60% for each of the individual markers, which is very different of a pattern than what we saw for the mates and the NKTs. And this is partially what we assumed was there's additional heterogeneity within the Vδ 2 group. We're just averaging across different cell subsets within the Vδ 2s, and this is kind of like what we end up seeing. And when I check TNF-alpha, as far as activations go, we have a similar pattern with both TNF-alpha and interfering gamma being anywhere from 30 to 60% of the cells responding and making those cytokines. So at that point, I checked with a marker co-expression plot, and sure enough, it does look like there was additional heterogeneity present. So for example, you can see like CD4 is pretty correlated with CD26. The NKG2D is pretty correlated with the CD8. So there's at least two additional heterogeneous clusters present that we weren't actually grabbing, and we're just averaging these together. Which brings us back to our second elephant in the room. We have all this data, we have all these markers. How do we leverage it in the most efficient manner to be able to get at the actual things of interest? So at that point, we had 3 million cells. It's relatively easy to extract out those little cord blood ILTs. But is there a way we could do this that makes it easier and more quantitative on our end, but also gives us the potential of looking at the rest of our sample? So at the central issue, the different clustering methods are tending to either inappropriately group or split things, which means you have to spend more time ungrouping them or resplitting them depending on what the case may be. Manual gating just does not scale, so it's not an option. I don't have that many lifetimes to devote to it, so it just was not possible. And automated gates are a good idea, but they just weren't easy to adjust in a way as far as the manual gates are. And then it also still remains unclear what drives the individual unsupervised algorithms that are oftentimes adapted for machine learning or other contexts. So what ended up being a 3 a.m. thought a little bit later after Luciana package was evolved was what if we had a way to combine the best features of both manual gating as well as the best features as far as being able to process large amounts of data that we see in the unsupervised analysis. And basically what I end up doing is I'm particularly interested in this point of the spectrum here, where I don't know how to rationalize where a cell population starts and begins if they don't share the entire set of markers. But what happens at the very end when you have terminally split everything and those cells share expression of every single marker? Is it actually relevant? That's a good question, but I can at least envision what those cells are as far as how they behave compared to a different cluster. So at that point, this is where I started working on a Core package, which started up you can see right around late October is when I first started actually working on that. And this continued through for early 2025. The basics behind the scenes working of this package is I'm leveraging those automated gating systems to at least do an initial guess as far as where the positive and the negative populations split apart, which I'm calling the split point. And then at that point, I'm doing more manual style interventions of applying gate constraints. And if it's still not cooperating, opening a little shiny app to apply the correction to ensure that all these potential divisions between positive and negative cells has been worked out for all these markers. At that point, what ends up happening is that gating information that summarizes the identity of the individual cell just gets rolled into a new column that gets placed within the FCS file, which also gets placed in the description keyword list, so we're able to retrieve it after the fact. And then what this really does is, beyond the analysis portion, is these unsupervised algorithms, regardless of what they're doing, they can still function perfectly normal for what they're doing. But then at the very end, I can retrieve and reaggregate and re-enumerate out the cells that ended up in a particular location or in a particular cluster to see if the assumptions as far as what we did by manual gating are the same as what we see for the clustering based on MFI, or if they are different. So it's a way of at least being able to check in on what previously had been a qualitative metric and try to make it a little bit more on the quantitative side. And we ended up doing this for the different algorithms. So at least in the context of these cord blood ILTs, they seem to be pretty heterogeneous in terms of their co-expression of markers. So regardless of which algorithm we are doing, we're ending up with similar clusters across the board. So without going too far into the nitty-gritty of every single marker for all 5 of the subsets we saw, basically when you're gating on an area of interest of a particular dimensionality visualization, in this case, this was a pack map, looking at the areas of primary density expression. And then once I get in there, grab the cells that are present, re-enumerate them back to the individual, I can kind of do the same process of if I had FlowJo open and did a marker expression for a particular marker, I can rederive out the individual frequencies in a similar way that is within 1% of what it would have been by manual gating at that point. And then at this point, we start seeing the patterns reveal in with the Vδ 2 population. So as we have known from the literature, there are CD4 Vδ 2s. In this case, these were CD27 uniform, and then they were mostly CD45 RA negative, so meaning they were memory differentiated. They had a lot of CCR4, CD25, PD1, very similar to what the NKTs had. And when they were stimulated with PMA ionomycin, they made a lot of TNF-α and a good amount of interfering gamma. Vice versa, for the CD8 population, we saw those markers that were pretty high in CD4 are just not there for CD26, CCR6. We see NKG2D is very high. We see that they are mostly CD27, CD45, so they are memory naive. And we don't see much of that CCR4, CD25. And then they're making more interfering gamma than TNF-α when they are stimulated. For the double negatives, it's kind of similar to the CD8s, but a little bit more muted across the board, making a little bit less interfering gamma and being a little bit more differentiated, all said and done. And then another population we knew about from the literature was the T17-like Vδ 2s. These were pretty uniform in their expression of CD26, CCR6, CD161. They were primarily naive as far as their definition by CD27, CD45 RA, and then they had a lot of CD25 for what we were expecting, especially compared to the other double negatives for CD4 negative, CD8 negative. And these cells just made a little bit of TNF-α. They did not seem to make interfering gamma or CD107. And finally, because we were acquiring so many cells to be able to measure the very few MAITs and NKTs, we ended up seeing an additional population of Vδ 2s across the vast majority of specimens that was PD1 high. And this was a particularly interesting little subset. I don't entirely know what to make of it. It doesn't share a lot of the same marker expressions we see with the other Vδ 2 subsets, but it is NKG2D negative, it is CD45 RA, CD27 positive, and it has a lot of PD1. And it has a good amount of CD107A, which is the degranulation marker we measure for cytokine release, the cytotoxic granule release. All things and done, this is just looking at marker expression profiles across everything. the main goal of our study remains, is there a difference between our unexposed infants and the different HIV-exposed infants? So when we check just the general marker phenotypes, as far as is there a difference in marker phenotypes, we see that CD62 ligand is increased in the HU high for both the CD4 negative, CD8 negative population, as well as for the T17-like Vδ 2 population. And then there's a little bit discordance for CD161 and CD56. But all the other markers in the panel for all the different subsets of Vδ 2, we don't see anything statistically significant in terms of their marker expression. And then interestingly enough, when it comes to activation by cytokines, we saw a very interesting pattern in the sense that the double negative, i.e. those CD4 negative, CD8 negative cells for the Vδ 2s, ended up producing more, had a greater frequency of cells making interferon gamma and TNF-α compared to their unexposed and even their unexposed low counterparts. Now, as you may notice, there's still quite a bit of variability in this HU high infants, going anywhere from like 20 to up to 60%. We try to correlate this to any of the known metadata we had in our panel. We weren't able to match it to explain what we're seeing. My suspicion is there's probably some element as far as the maternal viral load decline to what we end up seeing for these cohorts. And likewise, we saw two final additional interesting patterns. If you looked at our CD4 Vδ 2s and our NKTs, we see that they're sharing a lot of the same phenotypic markers, and they're also making cytokines at similar levels to each other. The main difference, of course, being they are obviously expressing Vδ 2 for their marker versus the Vα24, J-alpha18. And then we saw a similar pattern when it came to those T17-like Vδ 2s, where it had a lot of the same expression as what we saw for those MAIT cells across the board. There was a couple additional differences, especially in the CD25 expression, but it does seem to be matching what we see. And both of these are just making mostly TNF-α as far as their cytokine production, which we can summarize as well in more heat map styles that make it a little bit easier to scan versus trying to figure out where that middle measurement is for everything. And there was a couple additional things that we covered in our paper. We saw some differences at day 17 after expansion in culture for the IL-2 BCG, where there was less interferon gamma being made in H2 high, and some spec-specific differences for different Vδ 2 markers. And most intriguingly overall, those same markers we saw differentially expressed in the HU high compared to the unexposed, we saw that same pattern repeated for the CD8 T cells as far as the entire adaptive group in that same well. And we also saw this increase in interferon gamma for the HU high present in the CD4 negative, CD8 negative T cells. So not the Vδ 2 subset, but the broader one that also includes alpha betas and potentially Vδ 1 T cells. In terms of future directions, this is the conclusion for the cord blood ILT story. We have chased this rabbit down the hole, and we have pretty exhaustively profiled all the data that we had available for them in the context of this panel. The study in broader context was still looking at the different subsets. So the work on myeloids, the works on alpha betas, some of that proxy measurements of the NKs is ongoing or on the wrap-up phase at this point. So there will be more HAU papers using similar methods on the way in time. And then there still remains on the plate the matter of looking at the B cells. So we'll see what can be done in that regard. Ultimately, my goal when I started working with this cohort is this is a unique cohort. We were not going to be able to recruit another cohort like this in the future. So I wanted to make sure that all the cells that we got, we were actually mattered and that we could actually profile to the extent. So starting off when I was like reading papers back in 2021, 2022, and realizing not everything has been worked out in terms of the unsupervised analysis, as much as different vendors tried to sell us on that impression. We are now at a point that as a lab working on our different papers, the semi-supervised pipeline that we have implemented is pretty well documented, is pretty robust, and is able to do what we need it to do for our spectral flow cytometry data. And similarly, as far as open science and open source, all the R package work I've done is not just going to sit in a drawer. It's available on GitHub, and both packages are on the way to go to Bioconductor, which is the main repository for the molecular biology style packages, but then also the two that are more niche as far as the instruments themselves will be going out to the CRAN repository around the similar time. And as it was abundantly clear to me in the last few years, what good is building tools in R if the community as a whole is not actually able to use it? So since February, I've been doing the cytometry in R course, both here in person as well as online. I am still very shocked that I woke up the next morning after posting it on LinkedIn and 800 people had already signed up. By the time we started in February, 2,000 people had signed up. So I hope at least everybody has some little sparks of even though like my first journey into R, it was like 2 years before I ever touched it again. Everybody may benefit for years to come from this one. And all in all, it's been a rather weird PhD. So COVID was definitely an oddball. I wasn't expecting that. But I ended up taking my data analysis skills and just basically grabbing all those records from iLab and seeing where all these 18-hour experiments spread out during the course of my PhD. And you can see where most of them happened. I didn't actually update 2025. But then you can also see where code kicked off. But then also it's not entirely fair because most of my writing of both the manuscript and my thesis also happened in Corto. So some of those green dots are just actually writing, believe it or not, obviously the case. With that, I would like to do some acknowledgments. First and foremost, my co-mentors, Chris and Kirsten, they have put up with so much things. They dragged me out so many rabbit holes repeated times. My English is not the best when it comes to writing. They had so much red ink pens that they went through in the course of both the manuscript and the thesis. It's not even funny. You can ask them about it later. I also like to thank all the people who have come through our lab. Hao Ting was a postdoc back when we started and taught me how to do flow properly. As well as Winter, who went through our section last year, and then all the different summer interns, as well as the rotation students for MMI. I'd like to thank the MMI program in general, both Brett Hassel and Heather, who were there during my first few years, as well as Eileen and now Bess in the later half of my PhD. I'd like to thank my entire thesis committee, as well as everybody at the University of Maryland Flow Corps, who not actually providing those very fun instruments would have not started this rabbit hole of things to prospect down. And then the more final one, as far as the individual ones, and this is where I do not cry. I would like to thank that pesky cat I have, which has been trying to murder me, but also tried to love me the entire time. So she's nearly blown up the house twice by turning on the oven and filling it up with gas. But then the other time, she's perfectly amenable and perfectly lovable. So I have a love-hate relationship with this Alana. I would like to thank my parents, my brother, who are not here today because they're still far away. I couldn't have done any of this without them. I appreciate them immensely. I would like to thank all the coffee shops in Baltimore, specifically Koba, Daily Grind, and THB. Also Sam's Bagels, because they have kept me going for the last year and a half of writing. So always a shout out to them. To my cohort, Laron, Rainer, and Alex, as well as our honorary MMI members, Garrett, Matt, and Kirsten, for the early times in core and keeping us sane. I would like to thank my more outside of work social networks, both the RPCV network for the Returned Peace Corps volunteers, the Canton Kayak Club, the Downtown Sailing Center, everybody who does effort to keep Baltimore's parks and recreations facilities always open and available, especially when you need to clear your mind. I would like to thank the Patapsco Valley 50K for throwing the most random, eccentric, and crazy 50K Ultra on Halloween weekend. They give you pumpkin pie and bacon, egg, and cheese bagels every 3 miles. It's great. Everybody should do it. And then, of course, my fiance, Daniela. The world became much brighter when I met you, and I could have not done this without you. And with that, I'll take any questions. Hello.
One of the issues when you do a lot of these simulations with rare populations where you can't really enrich, like outbreak samples that are in themselves where really separate them out and stimulate. How do you deal with the possibility that a lot of the stimulations you're seeing could come from one cell making exit? That those differences are going into the mix, right?
Yeah.
So because you have so many parameters, is there a way to build in kind of a correlative mix within one group, which was either your HEU or control? The relative abundance of a particular cell type can be an independent variable to see if X amount of the alpha 2, you're going to affect gamma from the alpha beta intercept.
Yeah.
Yeah.
So the entire section as far as correlations between the cells is something I've been mulling about, and I've tried to figure out a good way of tackling because we've already seen some effects, and anybody who's ever done cell sorting will see this. Certain populations will tend to die off just for like random reasons. So at that point, you are biasing your data in some ways as far as what's seen. And then similarly, if there's actually more of a network interaction, especially for response to generation of cytokines, if some of those get tinkered one way or another, like for example, if you think of the NKT papers for the T17 data, those have greater amounts of interfering gamma made than what we saw in our cohort. I think it was one of those islands being made as far as what they were seeing. So I don't have a good way of parsing it out right now at this point in time. One thing I have thought about as far as like we have been kind of limited as far as like being able to just look at frequency apparent for each individual population, how to actually go about and tie out these different, if you like terminally cluster these little nodes, how do we measure the nodes in a way that's rigorous and it's not just overinflating knowledge as far as like there's a single dot that throws off and everything and makes it statistically significant. So it's one of those areas I'm still trying to parcel out as far as the best approach to be able to handle.
The work is very extensive in making different atlases for different subsets. So I'm sure future studies will be held by all of them. I was thinking about two findings. So most of your results with DE, they are like production of things that higher. So that one is expected.
Yeah. So it would fall within line of what we had seen for that first set of papers that did see an increase in cytokine reactions. And for the little bit of day 17 data that showed the decrease after culture, it kind of aligns with what those papers saw. So it's one of those things that it would be fascinating to be able to like dive into those individual papers at more depth as far as like if it was just they didn't have enough infants in their cohort, or if they are also seeing more of this heterogeneous effect where they just had one individual specimen with more CD4 subset, and that was enough to throw it off. So it's one of those things that we are seeing the pattern, but trying to reconcile the previous data is probably more effort than it's worth at this point. But yeah, it is seeming to follow that pattern. The important thing is in terms of actual impact, we started the study and we were enrolling HU high infants fairly frequently, but by the end of the study's enrollment period, we had not enrolled any HU high infants in a long time, mainly because government policy had caught up in the clinic and most of the moms were catching their ART earlier on, and therefore they were not actually showing up at the antenatal care visit with viral loads. So it's definitely one of those situations that if it is a situation that even if the differences we see for interfering gamma do correlate to what actually happens after birth, because they're so the unexposed low are much more similar to the unexposed. The unexposed low are much more similar to the HU. It may not be that same kind of effect. It's going to be seen much more forward. But that's speculation as far as like, does the overall pattern we see in response to more of the viral side translate to the entire adaptive immune system or innate-like immune system? So it's more of a network effect at that point. So
then comparing Vδ2 and MAIT.
Yeah.
There I was wondering, were you expecting them to be different and they turned out to have the same pattern? So that means it's difficult to differentiate which cell is contributing to you.
So I don't know if I was expecting anything versus I found it very cool to see phenotype and function kind of paralleling each other on the different branches. They are not hard to differentiate in terms of our overall panel. Like I can easily tell what the Vδ2s are because they do have that Vδ2 marker. And similar for the V alpha 7.2 or if we're using the tetramer. So I'm able to delineate them out. I was more intrigued by how it seems to be, and this is also partially biased. We included markers that we thought would be interesting. There could be a dozen other markers that are very different between the populations that just make it completely look very opposite of what we have. But at least in the context of our 29 color panel, it was interesting that it wasn't necessarily convergent, but they both ended up in a similar location.
If there are no other questions from the general public, we can close the public session here and we will see you later For the committee members who are online, please stay online. Now we will open the private discussion. Thank you, everybody, again for attending.